To the Microbiome Center meeting. You almost look awake today. So I'm hoping that you will stay completely awake because I know that Charlie's going to give us a great talk today. It's not often we get the entomologists because the entomologists have their seminar at this time. That is one of the reasons why we are thinking about moving times or doing a survey. So if you have not filled out the survey, go to the Monday, not the Monday, different list that I sent out. Go to the newsletter. There's a link in the newsletter. You still have time today to fill that out. If you do not select the time that you prefer, you may not be able to come to the Microbiome Center meeting, right? So go in there, fill them out, tell us what you want, because we're gonna go ahead and look at those carefully later today. We were supposed to be cut off by now, but I'm gonna give you guys until the end of the day. Okay, close the business 5 o'clock. We're done. So the idea, though, is really to try to get some of the entomologists in as well. And if this is the right time, well, I'm going to beg Gary to do something.
Carol,
yeah.
Do we have the people in the entomology department getting that?
Yes.
Fill that later?
I
Yes.
sent it specifically to Gary Wednesday evening and said, I was supposed to meet him, actually, and we screwed up we can't. Department heads cannot schedule for themselves. He and I were trying to schedule a dinner date with our spouses for ourselves. We can't do that. We now have proof that department heads are incapable anymore. But now you didn't need to know that. Nonetheless, now everybody knows that because we're recording.
Hello, world.
Nonetheless, so I said to him, please send this out to your department, and I hope he did. I hope
He absolutely did, and I can tell you people have opened it up and have filled it out.
Right. So
at least
great.
2 people have.
If you're like, nope, this is the only time I can make, counteract that right now. Yeah. Okay, so we have one more announcement, and that is next week, an hour before this, we're meeting with students who would be interested in a microbiome club. And so graduate students, undergraduate students in this room, send them here. We would love to have people involved in getting that kicked off the ground. We've got some administrative support for it, so it'll make it a little bit easier push, I think, for the students to begin with. But if any students are interested, if any faculty member would be interested in being the club advisor, I'll be here to start and to help with that because I advise a club, but definitely looking for 1 or 2 faculty members to go ahead and take that on. So without further ado, hey Charlie, thanks for coming.
Oh, absolutely. Thank you everybody. Thanks for having me here. I'm particularly excited to be here because I don't see many of you, well, ever. I'm usually always housed up in ASI, and so this is really nice for me to bring out and talk about some of the work that we've been doing in relationship to our microbiome stuff. But it's also exciting for me because really I think that we're at an inflection point within this work where we're actually getting more microbial and adding complexity to it. So getting impact or getting input from a diverse group of people who are much more on the microbe end of things, I think could be particularly valuable. So I thought I would just begin by talking a little bit about me and where I'm from and my perspective. I'm actually from Pennsylvania. I'm native to the northeast part of the state. I went to a branch campus as an undergrad, and then initially as an undergrad, and then I transferred to main campus as a grad student. I got my undergrad in agroecology, which is now no longer exists at Penn State, but more importantly to all the plant pathologists out there, I am at least part of you guys, part of your group. I have a minor in plant pathology. I then decided to go to Wisconsin, moved west to the Midwest, and did my PhD in entomology in Wisconsin, working on forest insects. And then I was enticed to return, and I've been here for the last about 4.5 years. The systems I've worked on are pretty diverse. I've worked on a bunch of different caterpillars and beetle species, worked on Gipsy Moth, bark beetles, Asian longhorn beetle, and most recently fall armyworm, which is going to be the feature of today. And really what connects all these different systems is my interest in plant defenses. And this is really fundamentally what drives my research and what gets me into the work in the morning, is I'm really interested in how plants are able to defend themselves against insects. And particularly, I'm interested in what are the mechanisms by which plants are able to defend themselves, and how do multiple forms of defenses cooperate in their defense against insects. And within this context, I'm interested in how does the environment alter the phenotypic plasticity in plant defenses. And really, I think that this is why I'm here today, is this last piece, is because I think microbiomes have provide a great tool for modulating plant defenses. And I don't know, I mean, I'm not sure how much interaction, how much this group talks about plant defense, but I think it's particularly cool because they come in multiple forms, multiple shapes, multiple sizes. They could be trichomes that physically disrupt an aphid landing on a plant. They can be like resins coming from a tree which expunge beetles. They can be incredibly toxic, like in cardiac glycosides, cardinalides, and milkweed, and they come in different, and the chemistries are incredibly diverse and fun and interesting and exciting, and they have so many different weird interactions with not just insects, but also their microbes. And as I said earlier, I think that part of the reason why I've been, actually, should I turn on the light at all, or does everyone can see everything pretty well? I just have some SEMs later on that I want to make sure you could see stuff. I think microbes are particularly cool good tools to study phenotypic variation, and I think out of this group, a lot of you can appreciate this fact. We know that the rhizosphere, bacteria, and fungi can modulate how plants express defenses. That endophytic and epiphytic bacteria and fungi can also alter these interactions. That when there's a negative attacker, like a plant pathogen, that this can alter the expression of plant defenses. And then I'm going to argue today, and hopefully I'm going to convince you that insect-associated microbes can also modulate or influence the level of defenses or the magnitude of defenses that plants have against insects. So how I'm going to framework this is I'm going to go through a little bit of a broader example that I think a lot of people can appreciate in terms of what the insect encounters when it's potentially feeding on a plant. And I just want to have everyone imagine that you have a series of choices that your kid and your mom got you a bunch of cereals and you have a series of choices. And maybe you might choose this one or you might choose another one. And this one, I mean, a certain number of the bowls of cereal are potentially going to have a jagged metal crustio. And you can, I know there's like, I know there's might be 5 people who've seen this episode of The Simpsons, but. And that you decide to you select this bowl, you eat that crustio, and then you are suffering the consequences. And when you think about what those consequences can be, they can be, if you chew on that and it breaks your teeth, that could be damage to your mandibles, and that could impact your ability to eat more. That's a little bit collapsed, but you could have disruption of your mucosal layer, and more importantly, your epithelial cells, where this barb is, or the jagged metal crustio is physically disrupting your digestive system. And accompanying this, and again, I think this vermin in particular can appreciate the fact that that could lead to sepsis. And this last part has been historically underexplored in plant insect interactions. And again, by the end of the day, hopefully I can show that, I can convince you that this is a component, that sepsis is an important aspect in terms of addressing the magnitude of plant defense. So different forms of defenses that we know we have. One of the benefits that we have with plant insect interactions is that we have really well-defined relationships between what plants express and how they interact in the gut. We know a bit about chemical defenses that disrupt insect gut functioning. We know protease inhibitors, chitinases, and phenolics. All these can have differential effects when they're being ingested and they're reacting in the lumen. And on the flip side, we also have physical defenses. These are the trichomes. These are the abrasions that the food bowls can create when it's being ingested. And collectively, these defenses have negative impacts when they're in the gut. We have loss of digestive efficiency, so you're not going to extract as much nutrition from your food. You're going to have metabolic cost of detoxification. You have potential damage to the epithelial layer, in particular microvilli, and then you have disruptive of the protective barriers, which in our case, it's a parotrophic matrix. In us, we have our mucosal layer. In insects, they have this barrier called a parotrophic matrix. I'll talk a little bit more about what that's comprised of later. And today I'm going to focus on these two guys, which is the damage to the epithelial layer and disruption of the protective barriers. And the central theme of the day is that the insect gut, this is the conclusion. I'm going to tell you right now. The insect gut is rich in microbiota and can heighten the effects of defenses that are targeting these particular substrates in the gut. So I'm going to walk through some recent work that we've been doing with fall armyworm. This is a polyphagous, so feeds on multiple different hosts. Polyphagous noctuid. It's a lepidopteran. It's a caterpillar. It's a moth. It feeds on it's native to the U.S. It feeds native to the U.S. It feeds on a bunch of different substrates. One of its favorite hosts is corn. Corn is not universally susceptible. There's a gradient and resistance in what they can do and the defenses that they express. And we're mostly going to talk about gut bacteria. So for the rest of the talk, I'm going to just tell you a little bit. There's going to be three parts to my talk. First, I'm going to talk about bugs and talk a little bit about the microbial ecology, in particular the microbiome of these insects and how we're able to manipulate it. The second one is I'm going to talk about how defenses can take these otherwise benign microbes and turn them against the insect and actually induce sepsis. And the third part is we're going to talk about how I'm adding ecological complexity to these already complicated interactions and potentially where we're going to go from there. So without further ado, I'm going to talk about bugs and bugs. The first thing I thought to do is talk about the history of sort of the interactions we have in plant and insect interactions. So insect microbe interactions have, I think we're sort of at a weird place within our subfield. And part of it, we're in a weird place is because we have this tendency to look at historical examples as guiding us for the new things we explore. And so so I'm just going to talk about these few examples. Aphids possess a symbiote called Buchnera. This is a really intertwined interaction where there's aphid and Buchnera. It provisions amino acids. There's this crosstalk in the metabolic pathways. It's a really defined one-on-one interaction. Cassida stammer, this is a leaf beetle. It has free-living bacteria that produce a pectinase. It is a one-to-one interaction that they occur through generations, transmitted from maternal to offspring. Termites have this complex of microbes that's being transferred between nestmates, meaning that they are performing anal-oral trophallaxis, and this microbiome is being passed between the nestmates and through generations. And they all have key physiological roles. This has nutrition, this has amino acids, this has these pectinases, and this has basically is able to take a block of wood and turn it into biomass, like insect biomass. And I want to bring up these examples and just talk about this because I feel like this is me thinking and me maybe I'm wrong, but this has colored a lot of the interactions as to how they're being explored now. Like these are clean, tidy, neat, clean interactions where you're finding the stability and you're finding these transfers either among conspecifics or between conspecifics. So what about the study system I'm going to talk about today, fall armyworm? And it's something different. And I'm just going to highlight a paper, Caterpillars lack a resident gut microbiome, and the accompanying nature news piece. Curious case of caterpillars missing microbes. And I bring this up not because this paper is a fine paper. I agree with most of the data, but I disagree with the conclusions. I disagree with the conclusions in the fact that because it's not that caterpillars microbes are missing, it's that we're using words differently. And this is, I think, actually like a kind of this particular news piece is actually kind of dangerous in terms of how the science being proceeded, the science is being approached, because this is a comment I had at a meeting this year at a Gardner Research Conference from a tenure track faculty, where, why are you working on caterpillars? Don't you know they don't have microbiomes? And my response is shrugging my shoulders and saying, I'm not sure you know what that means. Or how are you defining this? And really it boils down to is how this particular paper and how this news piece is defining resident microbiome. And they're defining resident microbiome as though it's occurring and propagating through generations, and microbiome being a stable set of microbial community. And another colleague of mine said this at Ecological Society this past August, is like, it seems like we're using the same words to describe different things. And we are. The way I use resident microbiome is that resident is you reside in where you are. And microbiome could really be anything. It could be variable, it can be stable, it can be whatever. Those are the microbes that are present. And maybe that's incorrect. Maybe I'm using it incorrectly, but that's the way that I've sort of been trained. And that's the way I'm going to be using it today. So a little bit bigger picture about what's actually happening in these guts. And I think there's a few key things to know. First is that in most cases, they are not stable and transmitted through generations. So you don't see this propagation of a single set or single suite of microbes through generation, in most cases. In most cases, some variation in the caterpillar microbiome is driven by the environment, and in most cases, diet, where we're finding that when they feed on different food sources, this is changing the microbial community. In most cases, these communities are very simple. There are about 10 dominant members, usually fewer. Usually there's maybe 1 to 3. And these members can vary. In most cases, many of these members can be obtained in culture. At least as far as we know, we actually have been pretty successful in culturing most of at least what lines up with our I'd say our 16S data sets. And in general, this is across all these, and I think this is a factor of just these issues, is we actually know really little about the physiological interactions in this huge important set of insects about what's going on between the insect host and their microbial community. And the reason I say in all these is most cases I tend to be conservative in nature by my own nature, and I'm pretty sure that there are exceptions to all of these rules. It's just that we haven't found them yet. So there actually is debate within my subfield about whether or not people should actually study this. And I argue, yeah, you should, because it's a hugely important group of insects and we don't know anything. So that's why I'm still studying it. So I'm going to talking about my system in particular, I think that the microbes or the microbiomes in these systems, my systems fall similar to these other lepidopterans or caterpillars that have been surveyed. This is my single MDS plot. This is the only 16S data set you'll be seeing today. Everything else is going to be through manipulations. What this is that basically highlighting that there are essentially two different groupings, where we have a soybean group, where the insect's feeding on soybean, it forms one clade. If it feeds on corn, it has another. And when we actually look at the members that are comprising these microbiomes, it follows similar trends as to what we find in other lepidopterans. We find a lot of Enterococcus, Klebsiella, Enterobacter, Pentoia, Pseudomonas, Scartobacterium. And this is to mention that we basically did the 16S data set, and we also, when we did the 16S assessment, we also curated a culture collection. So we have about 160 cultures in our collection to mess around with. This is actually funny enough, this is important, is that there's a positive relationship between fall-arming growth and the bacteria that are inhabiting its gut, which makes sense. Bigger the insect gets, the more microbes it possesses. Again, the reason I'm showing this is because apparently not all insects follow the same trajectory. If we take these data and we plot them on with other animals, so this is a paper that was published in these data are from a paper that was published in Proceedings from the Royal Society, where they looked at a bunch of different animals, like vertebrates, invertebrates, at different scales, and they made this nice little plot. And when we add ours to the data, it follows nice trends. So what it just tells us is that these are laboratory reared insects that are feeding on plants, and they follow similar trajectories to what we expect in nature. As they get bigger, they have more microbes. Another thing to know about the system is that when we typically feed our insects on artificial diet in the lab, and when you look at what's in corn, what you find massively different communities. And if you actually look at this is Enterococcus, this is one of our dominant members. When they feed on diet, it tends to be mostly Enterococcus. When they feed on plant, that could be more variable. And the flip is for Enterobacteriaceae. And the reason I mention this is that because laboratory cultures are almost always reared on artificial diet, and I think this is potentially biasing in multiple regards. First, it's biasing in the fact that the insect is feeding on this unrealistically high nutritive food source, but also the fact that we're biasing its microbial community. So if you actually go and you pick up an entomology journal, that entomology paper that looks at plant defense, what you'll find is that you'll usually find that the individuals reared their insects to a certain growth stage on artificial diet and transfer them to the plant. So what you're getting is a truly artificial system that doesn't have its microbes that would be in nature. And so this basically this is a problem. If you actually want to address ecological complexity and microbiome interactions in systems, I think you need to really bridge out and look at what's happening in the field. If you look what microbes are present in field collected caterpillar, they're incredibly different than in laboratory reared. So my question is, well, how do we deal with this variable community and dietary altered microbes, and whether we can really implement azenic and notobiotic rearing methods into the system to be able to tease apart these plant and microbial components. Sorry. And so what we did is we developed sterile rearing methods that basically we can figure out a way of manipulating the system. And knowing that sterile rearing methods for other animals is really expensive, we can do this we can probably make 1,000 sterile insects for $50 and a cup of bleach. It's really quite simple. We take bleach, about 5%, we toss our eggs in it, we agitate it, and then we put them on autoclave artificial diet. That creates an animal that has no detectable microbes. We then can take different bacterial components, whether singular or multiple, and we can reintroduce these to the animal by just applying them to the sterile artificial diet. And this is where we, I'd say, almost got lucky. Because what we did is we realized that we can go to the radiation facility down the street, and we can take corn leaves and we can gamma irradiate them, which sterilizes them. And so we have a system that we can manipulate what's associated with the insect and then apply it to a sterile plant. And this, I think, opens some doors. This does not work with all, I'll tell you, this does not work with all plants. We tried it with tomato, it didn't work. It basically fried it. It cooked it, didn't look healthy. It didn't work really well with tobacco, but it works really well with corn. And this is a show of what exactly is going on. This is uninoculated. So this is insects that are sterile. These are third instars, so this is the growth stage of the insect. These are colony forming units of these different isolates that we inoculated into the insect. And then when we rear them to fifth instar on maize, which is about 4 or 5 days, what we find is a propagation of the community. So the single inoculation is actually establishing, colonizing, and then increasing with the size of the insect without having to repeatedly inoculate the host. And that we still find 0 CFUs or colony forming units associated with the insect. So I've been hoping and trying to find something we can manipulate like this for, pretty much since I started grad school. And so I think that this provides a set of tools that we didn't have previously that we can really do a lot of cool things with, and maybe possibly answer some of those physiological questions that aren't being answered yet. So the first thing I wanted to do was try killing my bugs. So this brings me to the second part of the talk is, how does the bacteria actually enhance the impacts of plant defenses? So I told you a little bit about the insect. Let's talk a little bit about the plant. So for these experiments, we used corn or maize. And what we did is we used maize lines or maize genotypes that had previously well-defined interactions with fall armyworm. And so we basically use this maize genotype called MP708, and expresses should be cysteine, sorry, protease that damages the paratrophic matrix. This is a well-characterized interaction across the last 2 decades by one of my collaborators. So talking a little bit about, I said I would talk a little bit about the parotrophic matrix. This is the parotrophic matrix of a fall armyworm. What this is the parotrophic matrix is that it is encompassing the food bolus. So much like we have a mucosal layer protecting our epithelial layer, this protects the epithelial, the larvae epithelial layer from the abrasions from the food bolus. This is comprised primarily of chitin and glycoproteins, and what this does is, again, it protects against abrasions, but it's also been documented to protect against infection, and particularly infection from pathogenic fungi, viruses, and bacteria. Nonetheless, there are things that can actually disrupt and pierce and break the PM. This is a trichome. This is a trichome in fall armyworm. This is a fall armyworm parotrophic matrix. This is a trichome, and you can see that it's disrupting that protective structure, and that which potentially allows, again, infection to occur. More dramatically and more violently, this is that protease I just mentioned. This is the type of damage it can cause. It can cause significant perforations in the parotrophic matrix that allows the food bolus to be, to be open to the epithelium. And here's another perforation, and these food boluses and the food bolus and the associated food and in this internal in this lumen can potentially have microbes. And this is an SEM that we took. You can see a chunk of food, Hansen on the chunk of food, and that's Enterobacter. So these are potentially providing these avenues for these infections to occur. So this is the experimental workflow of what I'm going to show you for the next few slides. We used corn grown in the greenhouse, and then we gamma radiated it at the center. We used second instar larvae inoculated with one of 3 bacterial isolates, or the azena control. We conducted these bioassays for about 6 days, and then the resulting experiment. Then I did repeat the experiment using larger larvae to look at some immune responses associated with the insect. So the first thing I want to show you is this is a susceptible maze. What you'll find is that it's lower, like all these bacterial treatments are lower than the azena control. So there's a bacterial cost with it being associated with susceptible maze. When you look at resistant maze, what we find is that these responses are heightened, and in some cases that they are elevated with the interaction of particular bacteria. So this white circle is azenic maze. This purple one and this green triangle there is Klebsiella, and this red one is Enterobacter. And the reason that we ended up using these three particular isolates in single populations is, one, is that we haven't really this has not been documented before, and 2 is that this represents a dietary microbe, and these 2 are commonly found in the field in about 50-50 ratios. I think more dramatically, though, this is larval growth. More dramatically than larval growth is larval mortality, where we see a significant elevation in the mortality driven by the presence of these bacteria, particularly Enterobacter elevates it to about 35% mortality. So what we're finding is that there is this interactive effect between fall armyworm, protoworms, and plant genotype, and this is all driven by the bacteria that is present, but also by which bacteria is present. When we actually look with larger larvae, we're able to bleed them. We're able to do some things that are actually pretty violent, pretty unsavory. But we can bleed the insects, and we could look at hemolymph bacterial titers. So this is with our Enterobacter. This is susceptible. This is resistant. And what you find is when you have resistant maize, you have a lot of bacteria that are actually entering into the hemolymph, which is their blood. And this is much higher than in the susceptible. Now, the question that was raised to me last week is, well, why is there some bleeding in the susceptible? The susceptible maize is the one that has the trichomes. So it's not like, even though it's susceptible, it still has potential damage going on in its gut. What I think is particularly cool is that in addition to the fact that we have these bacterial challenges that are occurring in the gut, into the exiting the body cavity and entering, or exiting the lumen and entering the body cavity, we're finding elevated immune responses associated with these. So this is phenyloxidase. This is just a general insect immune response. What we find is relatively low levels and no interaction between susceptible and resistant maize. When we looked at Enterobacter on susceptible maize, we see an elevation in the immune response. And when we look at Enterobacter on resistant maize, we see an interactive effect, meaning that there's more bacteria that the insect is going to have to deal with and potentially clear. Similarly, we find with cellular responses, so these are hemocytes, these are just general cells, these are defense cells that are present in the insect's hemolymph. Azenic, we find relatively few cells, but when we look at Enterobacter treated, we have an elevated amount, but we don't see an interactive effect between the genotype and the presence of the bacteria. So we actually repeated these experiments again with using different sized trichomes. So we looked at small trichomes and large trichomes along with recombinant maize chitinase to see if this is evident across multiple combinations of defenses. And what we find is that, yeah, there's actually an interactive effect. With azenic caterpillars, we find a negative effect of growth. The chitinase has a minor effect on growth. But when we actually look at the chitinase interaction with the Enterobacter, this is significantly elevated. Again, this is growth. This is with small trichomes, and both these treatments have Enterobacter present, and we see a stronger effect with the chitinase in the large trichome. So meaning that there's an interaction between the physical defenses, chemical defenses, and the microbial component. And similarly, we see elevated levels of microbial titers in the hemolymph. So one of the questions that we had was, well, is it just the fact that these bacteria are negative by themselves? So what we did was, again, we could do some pretty unsavory things. We can basically take insects and inject them full of, and try to make them something. So we took insects, reared them on artificial diet for 4th instar, and then we added PBS or phosphate buffered saline, Enterococcus, Klebsiella, and Enterobacter at 10 to the 6th cells. We've actually elevated, we went higher, like we went up to 10 to the 8th, and we got the same response. So we ultimately looked to see if we can basically inject these into the hemolymph and then look at growth and mortality. And we found negative effects of these injections on the growth, meaning that there's a cost associated with clearing these infections, but we never found sepsis. Like even at really, really high levels, these bacteria alone didn't incur sepsis. So like it's not like we're injecting something that's pathogenic. We're injecting something that's the insect's clearing. So here's our model. We have an insect, and we have resistant maize. There's direct effects of resistant maize. And these direct effects are related to having to repair the peritrophic matrix, which is costly, a loss of digestive efficiency, and they ultimately have reduced feeding. So these are, and again, this has been defined for this particular system well in the past. What we're adding to the equation are these indirect effects, where there's this indirect effect of having microbes that are invading and triggering immune responses, and the associated costs with either clearing those infections or potentially not. And the way I describe it is like, well, you have the way I kind of equate it is that I need a new roof, my basement's flooded, and my check just bounced. Is that you have a finite amount of resource, you have a finite amount of energy pool, and you got to deal with both these issues, and you can only, sometimes the stress is too much, they can only deal with one. So one of the caveats that we had with all this is that, and this is criticism that we received in the review process, and a criticism that I still have, is that we're mostly, up till now, I'm showing you work that's been done with populations. and which I think is a good starting point, but ultimately we need to add complexity to this equation, and we need to add more ecological complexity. Particularly, we need to try to mimic what is happening in field populations and try to understand what if these interactions are holding, or if they're holding, or if frankly if they fail. And this is my criticism. And particularly the questions I'm interested in: are there isolates that may amplify or dampen the effects of defenses in our system, and how do herbivores that have more of these field mixtures or wild mixtures may alter these equations. So this is what we're doing now, and this is ongoing work. So I'm going to show it's preliminary. So please don't skewer me. But this is also where I was hoping for, I'm hoping that people have input and feedback, because this is basically, the more I'm doing this work, the more I'm getting out of my comfort zone, which is a good thing because I'm growing. Alternatively, it's freaking me out. But but what we've looked at, what we did this summer is that we have this set of isolates. We have 160 isolates, and what we have available to us from in the past is that if you look old school, like last 30, 40 years ago, there's a lot of work was done with plating and with looking at screening virulence factors, and particularly looking at virulence factors of one insect pathogen called Serratia marcescens. And that these virulence factors can actually predict, at least for Serratia, was able to predict whether they are pathogenic in Drosophila. So what we did was we went through and we screened all 160 isolates across 7 different media. I had a very enthusiastic undergrad do that this summer. And so this does not distinguish between whether we actually have a 16S sequence of it. We just did every single one we had. And what struck me was the fact that, first of all, virulence factors across the board are relatively rare. That with the strains that actually but that we actually have a lot of phenotypic variation within our systems that we could mess with and play with. And so what we're starting to do now is just basically use this as a way of us selecting for a bacteria and feeding it to our insects and seeing how it performs, seeing how the insect performs. And can we actually find things, use this as a way of finding microbes that are potentially more lethal to our insect, which is ultimately what we want. I'm interested in killing things.
Can I ask a question?
Yeah.
So did you look at chitinases?
We did. So we looked at chitinases. Most of them can grow in chitin. Most of them are able to form cells and grow on chitin. The reason I didn't list it up here is because we actually didn't see, we used a colorimetric method to look at changes in terms of clearing. We actually saw very little clearing. When we saw, we saw maybe there might have been 6 or 7 isolates which could not subsist on chitin. I think we need to repeat the experiments and just because maybe I think I might have messed up. Maybe I messed up our colloidal chitin. But yeah, they can grow in chitin, but we haven't seen any clue zones of clearing. And
chitinase genes? We
haven't screened any genes yet. Yeah, so the current idea is we're going to try to get genomes of some of these too. So yeah, this is all we're all at a very low level right now. But I think that we're onto something with relationship to these virulence factors, because this is our bacteria that I showed you before. This is as you can see, that's a little bit heightened. This is azenic. Azenic is always, I've been doing these experiments for 2 years, and I'm really good at not killing azenic insects. But when you look at some of the other factors, we see these different responses. We see some that are really heightened, some that are follow the same pattern, and some that actually stay stable. And we really need to, we're just starting this. We're trying to figure out how we're going to do this, because we have a lot of isolates to screen, and how to do it in a way that's consistent and clean and reproducible. But I think that we might be on. I think that there might be some interesting, microbial ecology to do within this organism. On the flip side, so in addition to taking our current culture collection approach, what I did this last few weeks, and actually like I have an experiment ongoing right now with this, is trying to perform fecal transplants that are done in other systems. Or in our case, we have frass, so maybe frassical transplants. So what we did was I took lab insects and I gave them a field that was grown in the corn at Rock Springs. This is what the frass looks like. It's in a nice cup. Get some food, remove the food. Then you can make this frass slurry, and then do some different. Yeah, I know. Oh, it's great. Yeah, it's thankfully this shit don't stink. So one thing that we have going for it.
But this brings up a really interesting thing we could do in the Microbiome Center, and is that would be a poster of the most disgusting things that we do as microbiome scientists.
I've got.
Good, thank you.
But then we actually it's what I realized is that it's really easy to clean up bacteria or to do differential centrifugation within this system. Like, took us like maybe 1 or 2 tries and able to get a nice concentrated bacterial pellet. And then we give new recipients. So the second steps. So I'm just going to orient you to this x-axis first. So B73 is the one with the small trichomes, TX601 is the one with the long trichomes, and MP708 is the one with the resistant protease that causes all that proliferation. What you find is that azenic insects follow this trend. They perform less well on long trichomes, they perform really poorly on MP708. When your fecal transplants follow similar, yet more heightened trends with the long trichomes, and it tends to level out. We still need to do a lot of work with this. Again, this is something that's ongoing right now. In particular, what's interesting to me is that within both these treatments, what's driving a lot of this, like this downwards variation, is we find a lot of mortality. And the funny thing is something that I've been reflecting on the last few years is that if you're interested in killing insects and you're looking at or using microbiomes to potentially do it, we're sampling live insects. We're sampling the ones that made it. We're sampling the healthiest, most vigorous insects. You don't want to sample a dead insect to get its microbiome. Why not for us, why not actually take the ones that are dying and look at those ones? Because those might have the microbial players that we can manipulate and potentially enhance them. That's maybe a little bit too upward thinking, too hopeful thinking. But this is sort of what we're doing now. And I'm pretty enthusiastic about it. So again, I think that there's a lot of interactions that we can explore, and particularly like there seems to be, like we were discussing last week in an entomology seminar, whether or not we would see dampening effects of multiple microbes, enhancing effects between multiple microbes, and ultimately how context dependent some of these relationships are. So currently we have some proposals submitted to try to look at some of these particular interactions. But my concluding thoughts and hopefully take home messages, and hopefully at the end of the day I convinced you that microbes can play an important role in these relationships, in particular skewing and enhancing the magnitude of defenses. But ultimately it comes down to context and ecological complexity, and I think this is a no duh. It's that there is this tendency to simplify, and I think sometimes we need to make things more complex in order to really understand what's fully going on. Ultimately, the direction and magnitude of the interaction likely depends on the type of defense's presence and probably particular microbes. There's probably microbes that we're finding that there's probably ones that do a worse job in terms of enhancing defenses, and some who probably do a better job. What excites me, and again, like from my interest in plant herbivore phenotypes, is that I think that again, microbial variation may allow for these diverse host phenotypes. And I think that the more we explore these types of interactions, particularly in entomology, we're going to be able to find more. And what's interesting is that, and to me, is that one of the things that keeps on coming up is, well, we don't know if there's beneficial roles of these microbes. Like why are all these caterpillars as rich in microbes? They're costly. They're potentially going to be pathogenic. Like what is the ecological and evolutionary role of these? Is there? But I really do think that within the tools that we developed here, we have a way of addressing this. whether that's through differential gene expression or whether that's through metabolomics. I mean, we might be able to get to answering some of these questions. With that, I'm currently funded by USD and NIF, a postdoctoral fellowship. I really want to thank the Radiation Center Radiation Science and Engineering Center. They really opened the door for us to be able to do a lot of this work. I don't think if we didn't have their expertise and their patience and their flexibility, I'm not sure we'd be able to do it. I also want to thank, obviously, my college and all the imaging, all the SEM stuff was helped with. Missy and John helped with downstairs. I think that you're only good as the team that you surround yourself by, and I've got a great team. My postdoc advisor, Kelly and Gary, really supported and kind of let me drive this research in whatever direction I kind of see fit. EK is a pathologist, is an insect pathologist, and he really helped out with a lot of the, I think the frameworking of the initial projects. Asher, I want to highlight Asher and Ray for basically dealing with me on a daily basis, which is not that easy. And lastly, I want to thank my new undergrad, Abby. She's the one who did all the screening. It's just basically been superb. And I'm really, really impressed. With that, I'm happy to take any questions or feedback or input. So thank you.
When I think of the armyworm, I think baculovirus.
Mm-hmm.
Do you think that the different gut microbial constituents would affect the ability of that virus to infect and kill them?
Yeah.
Like positively, negatively?
Actually, so I have data with Gipsy Moth that shows that the presence of bacteria, like comparing azenic to like bacteria, or to having bacteria, the ones that have bacteria have enhanced mortality or higher mortality. So there's a trade-off between, yeah, I can't tell you why,
yeah,
but yeah.
So if I'm remembering correctly, you said that the direct infiltration into the hemolymph was not as damaging as the natural infection. So do you think that there's something about the natural infection route that makes the bacteria potentially more virulent, or do you think it's just that combination?
I think it's the combination because, again, ongoing things I didn't show you today. We take that one that had really heightened mortality, you inject that one, they die pretty quick. So I think it depends on probably the virulence of the actual bacteria. Now, I think so. Yeah, to answer your question. Yeah.
Yeah, I was just got to say, it's nice basic study, fans, beyond mammal diet, but due to the practical side there, it's already known that we have a really effective bacteria that occurs.
Correct.
And then it occurs via sepsis.
Yeah.
And it doesn't work in the thermal situation because it's expensive. So what makes you think you're going to find something that will work better than DP? And Don already knows.
What? Oh, I know.
Don already knows.
Oh, I know.
DP toxins are intense. So, I mean, I don't get it. I mean, you really think you're going to find something better than DP? I mean, and we don't have to modify.
They don't have to modify. I don't know, but I mean, but I ultimately I don't know. Also realizing the fact that, well, I'm not sure. Well, I know Bayer and DuPont and Monsanto. Well, it's historically, I'm not sure who owns who now, but they've opened up like microbial like discovery people to look for other things other than BT. And my stance on that, relationship to finding if it's enhancing BT is that it's not so much of finding something that we can apply to the plant, more so that can we think about finding something that we can deliver along with the plant. That's not just a gene or not just a set of genes, but can we take the microbe and make it basically create endophytic or make it endophytic, and can that enhance resistance? So less of finding like I'd say less of finding like a BT analog and more finding like a BT complement. I'm not sure. Ultimately, you're completely right. BT's everywhere because it's yeah.
But it doesn't really work to control high populations.
Yeah.
But I would agree with you that you know, where your work is leading and where it can lead others is to really look at plant resistance and how microbes delivered by insects or that are there and taken up by insects through the other. What is there that is enhancing resistance?
Yeah.
And that's what that's another avenue.
Yeah.
Salary.
Well, microbes and insects actually have a negative effect on the caterpillar because, you know, they're going to induce toxic acid. Boundary. The AC.
There's complex interaction. Yeah.
So I'm not an entomologist or plant scientist,
Yeah.
but when I think BT, I think through the food science part of things. This is a spore-forming bacteria that's much harder to get rid of when you supply. And the bugs you're working with here are pretty wimpy in terms of what we use when we process our food in order to get rid of it. So if you think from that perspective, and it's not that expensive, like if you look at spraying with BT or another bug that gets wiped out with very short pasteurization, you're better off using these new bugs instead of BT because you know you're going to kill them. So there is another avenue that would be going after there.
Okay. Yeah, Daryl.
So these bacteria that you're finding naturally present, how specifically adapted are they to our herbivores? Or could you?
Can we swap them between different species?
Exactly.
Yes, we can.
What if you competed them against each other? You have existing ones, and you take ones isolated from some other insect and put them in with the ones that you isolated here. Are the sort of native ones going to outcompete?
It's a great question. We're not sure yet. Right now, waiting on data to get back related to, somewhat related to that question. So how does the host environment shape these, or shape the mixture of
Like, and I think that maybe gets to the point of whether it's a true microbiome or not. We expect some sort of interplay there where the host is shaping it, being adapted to that in some way. And that these aren't just transient ones that happen to get in there because they were on what the insect was eating. They are actually somewhat adapted to that.
Yeah, it's interesting to me. It's interesting because that line of questioning is interesting. Part of this is because the environment of these guts can be extreme. These are pretty alkaline guts. These are actually pH of 10. Yet they're still finding this. Again, it's not like they're clearing through at every single bite of food. I mean, one inoculation, they're established in there.
Yeah, I think that was shown pretty nicely when you have like the sterilized bees and putting these guys in there. They're not clearing them out; they're actually increasing the numbers. So that's pretty good line of evidence to me. But I'm also curious as to whether other ones from other insects do just as well, or if these ones have some sort of.
No, I mean, we haven't actually. I just transplanted some of the Enterobacter we have we collected from this bug to another species, but we haven't done, or another member in the same same family, but we haven't done like our cross families yet. We haven't done any of that stuff, but seeing if there's a basal trait of the microbe is something that I'm particularly interested in looking at. Yeah.
So what is your IRB for injecting insects?
I don't think we don't have one. I don't know. I or what?
Okay.
This is-
We'll cut the.
Mm.
Insects are not considered animals.
Is that
Yeah.
right?
I
Yeah.
doesn't care about
Okay.
it doesn't have
Interesting.
Yeah, we can do like yeah.
Well, I'm sure, I mean, each one of your advisors should definitely.
Only vertebrates.
Yeah.
But they all say, as far as I know, I don't be only.
Interesting.
Mhm.
There's
I
some
mean,
people
not
that
that
like
blind,
to extend to that.
I just wanted to say that the way you like made your own notobiotic like acidic bugs is amazing. I'm just so dancing on my chair back here. Did you get any mortality when you started bleaching them? What concentration did you use?
So yeah, the eggs are pretty fragile. So for this insect, we basically can max out at 5% bleach, and that's about enough. I mean, we basically put it. It's really easy. We put them in a beaker, spin it around with bleach, rinse it with water. We're good.
When you're feeding them, do you keep them in like a closed
Yes.
compartment, or how do you keep them from being inoculated by just the air?
We do everything in a biosafety cabinet. We're basically really that's all we really need to do. I don't know, just really clean. Clean going in, clean going. I mean, we don't need to. We're not like our rearing facility. It's not even a facility. It's like our normal biosafety cabinet hood, doing everything under filtered air, being mindful of what we're doing. Like we don't seem to have an issue with microbes coming in from air for our systems. Or we haven't. There's been a few cases there. I'll say we do periodic checks through our rearing protocol. Occasionally, if something goes bad, we just toss the experiment and start over. But some of the other insects that we work with, I mean, there's another one that, I mean, we could bleach it for 10 minutes we could bleach it for 10% bleach for 10, 15 minutes and hatch fine, they perform fine, they just do their own thing.
Another
Yeah.
question. So your heterotrophic matrix.
Yeah.
The native microbiome bugs in there, are they feeding on that at all? Do they produce their own proteases or chitases?
As in the microbes?
Yeah.
Yeah, so they have their own proteases, they have their own chitinases. We have no idea. And that's kind of why I think I'm so exciting. It's like you immediately think, for these groups of insects, guess how many metagenomes have been published? 1. And it was published in 2010. And what they did was they cultured everything and then they washed the bacteria off the plates. So that's the level of so we're still struggling with what does this variation mean? And that's sort of why we have very little, we have not compared dental gene expression between azenic and notobiotic. We have not done that yet. And we're really building from a low, I'd say, level of knowledge. That said, we can look at Drosophila and use that to generate hypotheses. But the problem with Drosophila and comparing that with these insects is that their guts are so different, their feeding behavior is incredibly different, and their diets that they interact with are just night and day. So I have a hard time extrapolating what's happening in those systems to these without really making all those caveats. So yeah, that's sort of.
Is it known how dynamic? Think of our mucus layers, they're pretty dynamic. We're constantly swapping off something.
Yes.
More permanent, or is it still dynamic?
It's dynamic. Yeah, it's being secreted. Here's how it can be complicated. Is that where it's secreted in the gut, can vary. So some of them secrete it like right when food is entering into their digestive system and then passes all the way through. Some secrete it along along different junctions along the gut. It really varies depending on the species. So that mileage may vary depending on microbial interactions, which makes me wonder about specificity.
Well, thanks so much, Charlie.
Yeah, no, thank you.